Welcome back on my channel. So now I'm continuing, biomolecular basis of inheritance, and subtopic is the search for genetic material, right? Okay. Even though the discovery of the nucleon by the Masker and the proposition of principle of the inheritance by the Mendel were almost at the same time, by the DNA act as a genetic material look long to be discovered and proven by 1926, right? The quest of the determine the mechanism for genetic inheritance had reached the molecular level. Okay. Previous discoveries by the Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and the numerous other scientists had narrowed the search of the chromosomes located in the nucleus of the most cell. But the question of the what molecular was actually the genetic material had not been answered, right? So we will continue. Transforming principle. In 1928, Frederick Griffith, in a series of the experiments with the Streptococcus, right, pneumonia, pneumonia, bacterium responsible for the pneumonia, okay, witnessed a miraculous transformation of the bacteria. During the course of this experiment, a living organism had changed a physical form, right? When Streptococcus pneumoniae bacteria are grown on a culture plate, right? Some produce a smooth shiny colony, no problem, and have a mucus polysaccharide coat, while R strain does not, right? Mice infected with the S strain die from the pneumonia, right? Infection, but mice infected with the R strain do not develop pneumonia. See, given है S strain है, R strain है, तो S strain injected into mice die, and R strain injected into mice live. ऐसा समझ में? मतलब अगर आर स्ट्रेन अगर उसको लगाया गया इंजेक्शन तो मर गया, लेकिन अगर एस स्ट्रेन, लेकिन आर स्ट्रेन जब लगाया गया इंजेक्शन तो जिंदा रहा, राइट? Griffith was able to kill bacteria by the heating them. He observed that heat killed S strain bacteria injected into mice did not kill them. When he, अब आगे वापस से S strain heat killed मतलब इंजेक्टेड इनटू माइस लाइव, ओके, एस स्ट्रेन हीट किल्ड प्लस आर स्ट्रेन लाइव, तो ये इसमें क्या है, जब इसको इंजेक्ट किया जाता है तो वो मर जाता है, राइट? इम्पोर्टेंट है, इसको ध्यान दीजिएगा, ओके. सो नेक्स्ट वी विल सी इंजेक्टेड a mixture of the heat killed heat killed S and R live R bacteria, S और R bacteria, the mice dead. अगर एक साथ अगर दोनों ये भी लगा रहा है तब भी ये मर जा रहा है. Moreover, he recovered living S bacteria from the dead mice. मोरेओवर समय उसने क्या किया, कुछ को रिकवर कर लिया, right? So next we will see He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide, okay, coat and become virulent, right? This must be done, due to the transfer of the genetic material. However, the biochemical nature of the genetic material was not defined from this experiment.